Hello, everyone. My name is Matthew Garnett, and I'm a senior group leader at the Wellcome Sanger Institute. And it's my pleasure to give this seminar today. I would like to thank the organizers for inviting me to attend this meeting and to have the opportunity to present. So very briefly, these are my disclosures. So genomics-guided precision medicine is increasingly used to treat cancer patients. And this can lead to more effective treatments and also decreased toxicity for patients. And so in this depiction of the slide, a molecular diagnostic is performed on a patient sample that will really inform on the molecular makeup of that patient's tumor to help inform the best possible care for that individual patient. And today, many precision medicines are based on the concept of oncogene addiction, whereby a drug directly targets a dominant acting oncogene or pathway that's selectively required for the survival of cells and their proliferation. So an example of this approach has been the development of increasingly potent selective EGFR inhibitors for the treatment of EGFR mutant lung cancer, as shown in the picture. Another approach is based on the concept of non-oncogene addiction, whereby the molecular changes that occur in cancer cells create a liability that can be selectively targeted in the cancer cell. The difference here, though, is the target itself is not oncogenic. And this approach might be particularly attractive for targeting tumors that lack an obvious dominant acting oncogene or an oncogene that is currently undruggable. So examples of this include targeting the enzyme PARP in cancers that are deficient in homology-mediated DNA repair processes, such as BRCA-deficient cancers of the breast and the ovary. And treatment with a PARP inhibitor leads to the accumulation of cytotoxic double-strand breaks in these tumors. A more recent example is due to the increased mutational burden of mismatch repair deficient cancer. So these are cancers that have a deficiency in the mismatch repair DNA pathway. This creates additional neoantigens which are recognized by the immune system. And through the use of checkpoint inhibitors such as anti-PD-L1 or PD-1, you can actually block the ability of the tumor cell to evade the immune system, leading to selective immune cell mediated killing, tumor cell killing. And of course, underpinning this is this pre-existing deficiency in mismatch repair, which creates this dependency that can be targeted through this non-oncogene addiction paradigm. And so now there's an increasingly large arsenal of precision medicines approved for clinical use. This table comes from a review I recently wrote, just looking at FDA-approved drugs for which we have a genomic therapeutic biomarker that allows for patient selection. And this encompasses 13 different tumor types. Most of these drugs are based on this concept of oncogene addiction. So they directly target an oncogene in the cells. Interestingly, some of these drugs are actually effective in more than one tissue type. So they can kind of be redeployed into different tissues that have the signature or marker therapeutic biomarker. And I think really importantly is to note that some of our most effective therapies work in relatively small percentages of patients from a particular indication. So in some cases, for example, patients who have mutations in the gene ROS, this can be down to less than 1% of patient populations. And the molecular heterogeneity of patient tumors and their variable response to drugs often makes developing these types of precision medicines particularly challenging. And so while these are really important clinical successes, even when using some of our most advanced medicines, it actually remains quite difficult to predict who will respond to a particular drug. And unfortunately, today, the majority of patients still don't benefit from this type of precision medicine approach, really arguing for the continued development of better treatments for the majority of patients. And we know that drug development in oncology is extremely difficult. And historically, success rates for oncology drug development are actually less than 10%. And that's actually shown here, looking at the success rate over time from a phase 1 trial to clinical launch of a new medicine. Fortunately, though, actually there's increasing evidence that genetically informed targets are more likely to proceed through each of the phases of drug development. And this is shown here in this table, just looking at success rates and the odds ratio for drugs that have genetic support for a particular target versus those that don't. And when you look across all stages, what you see is that drugs that have this genetic support are actually more than twice as likely to actually result in the successful development of a drug. And so from an industry perspective, increasingly the emphasis is on the development of medicines that are really guided by this deep genetic understanding of the disease. So to facilitate precision medicines, my lab for many years has studied how the features of cancer cells impact on disease mechanisms and therapy response. And we've been very fortunate over a decade or more to build up what I would describe as a preclinical discovery ecosystem to achieve this. This includes the ability to very efficiently make new cell models of cancer, the tools to do deep genetic analysis of those models, as well as large-scale functional perturbation platforms, including high-throughput drug screening or testing, as well as increasingly the ability to perform CRISPR screens very efficiently. To analyze these data sets, we develop new tools as well as web tools to really share these data sets to the community. And ultimately, what we try to do is develop mechanistic studies to really understand the observations we make in these models from these screens towards developing new therapeutic hypotheses that can hopefully translate into the clinical setting. So as part of this work, we've recently launched the Cancer Dependency Map initiative. And this aims to use large-scale functional genomics approaches to identify vulnerabilities in cells, what we call dependencies. And we aim to do this in every cancer cell. And we believe that a comprehensive map of cancer dependencies and the biomarkers that predict them could really help to serve the community by accelerating the development of precision cancer medicines. This is an ambitious project, and it's a collaboration with multiple partners in the US and Europe, including the Broad Institute, as well as our neighbors at the European Bioinformatics Institute here in Cambridge. The Sanger Institute's effort is actually composed of 4 parts, and we think these are the kind of key elements that we need to deliver on a dependency map. The first is models, which, as the name explains, is really about making new and better cancer models. Drugs is about perturbing cells to drugs to think about how we can use drugs more effectively. Genes is by perturbing genes to identify new targets. And of course, we need to build new analytical tools to really interpret and use this data. And so to assemble a dependency map, a key is that we're using very large panels of cancer cell models together with functional genomic approaches. For example, drug sensitivity testing. And we use these data in many ways to study cancer gene function, to discover therapeutic biomarkers, and to identify new drug targets. And to date, the Sanger and Broad Institutes together have actually generated data for more than 1,900 distinct cancer cell models, including things like genomic data, for example, whole exome sequencing, epigenetic data looking at things like DNA methylation, increasingly proteomic data, as well as now functional data sets such as drug testing and CRISPR screens. And coverage of the cell lines is actually shown in this image here. So you can see it's a very extensive data set. And at the Sanger Institute, historically, we've really focused on a set of approximately 1,000 models that we've very, very deeply analyzed and phenotyped. And I really want to emphasize that we use this large collection of cancer models to have representation of different molecular subtypes that occur in cancer patients, and that we know underpin differential therapy response. And as I'll show in my presentation today, we continue to expand these data sets through the development of new cancer models and their enhanced annotation with new molecular and functional data sets. Many of the data sets that I'm going to talk about today in my presentation are available through data portals that are hosted by my laboratory, allowing you to access, mine, and visualize those data sets. The first is what we call the Cell Model Passports, which is a hub for information about preclinical cancer models. The second is Project SCORE, that hosts our data for identifying dependencies in cancer cell lines through CRISPR screening. And finally, the Genomics of Drug Sensitivity in Cancer portal, where you can find information about therapeutic biomarkers to drug sensitivity. And these data sets are widely used, and we encourage you to have a look if you haven't done so in the past. So an important component of this work has really extended now almost over a decade, has been the use of very high throughput drug sensitivity screens across this heterogeneous collection of cancer cell lines to identify markers of sensitivity. So as an example, the data shown here are the IC50 values of 903 cell lines screened for their differential sensitivity to the BRAF inhibitor dabrafenib. And what we've measured here is the effect of dabrafenib on cell viability as determined by measuring an IC50 value. Each circle here represents an individual cell line. And the real take-home message here is just through this one example, you can see actually the range of sensitivities to this drug across this diverse set of cancer lines. And of course, what we can then do is we can integrate this drug sensitivity data with the genomic data that we have for the cell lines to understand the molecular changes that underpin the range of drug sensitivities we see. So you can see in this example on the very left, this is looking across all tissue types together, is that BRAF mutant cancer cell lines have an increased sensitivity to BRAF inhibitors. And in fact, we can see that if we look specifically in melanoma cell lines, this effect is even more pronounced. And of course, we know that BRAF inhibitors are clinically approved for the treatment of BRAF mutant melanoma patients. Now, in contrast to that, NRAS mutant melanomas are actually quite resistant to dabrafenib. And again, this is consistent with what's being observed clinically. And then finally, and I think most interestingly, is that precisely the exact same mutations in BRAF occur in the setting of colorectal cancer. However, unfortunately, these patients don't benefit clinically from BRAF inhibitors. And indeed, that's what we see in our data, is that colorectal cancer cell lines where the BRAF mutation are much less sensitive to the BRAF inhibitor than melanoma cell lines. And so I just show this example because I think it demonstrates how using these very large scale screens and in an unbiased way, we can identify clinically relevant markers of therapy response. And they also show the impact of both molecular background and in some cases, tissue lineage on shaping response to therapy. So beyond that specific example, we've developed methods to integrate very large-scale genomic and drug sensitivity data sets. And to date, we've actually screened full dose response curves for over 1,400 different anti-cancer drugs, generating over 1 million dose response curves. And so in the study shown on this slide, we actually correlated the sensitivity of hundreds of different drugs with cancer drive mutations, identifying hundreds of putative biomarkers of drug sensitivity and resistance. And the volcano plot shown on the left-hand side really depicts this and shows biomarkers of drug sensitivity and resistance. So each circle represents a statistical association between a genetic driver event and differential either sensitivity or resistance to a cancer drug. We can look at these examples in more detail, as shown here in these scatter plots. For example, in the setting of lung cancer, we can see that cell lines that have a mutation in EGFR are really sensitive to the EGFR inhibitor gefitinib. Interestingly, in fact, we know that clinically you can observe resistance to EGFR inhibitors through a coincident T790M mutation. And in our cell line collection, we actually had one cell line that had this coincident mutation. And indeed, as is observed in patients, this cell line was also resistant to EGFR inhibitors. In addition, this approach identified many new biomarkers of drug activity. Now, a challenge that we face at the moment is many of these are really quite poorly understood with our current knowledge. But nonetheless, we think that these could contribute to the development of these specific drugs, as well as new drugs to these targets. And so this is a very rich resource for developing new biomarker hypotheses for drugs. There are also, though, many unexplored potential cancer drug targets. And of course, for most proteins, we don't have a specific drug that can be used for this type of screening to evaluate the suitability of a therapeutic target and in what context it may make a good target. And so the recent advent of CRISPR screens provide us an opportunity to identify many new targets in a way that was not previously possible. And so to do this, we've really exploited the programmable nature of CRISPR-Cas9 system to investigate gene function in multiple cancer types. And this is something we call Project SCORE. So just as a bit of background, to find these new drug targets, we've been performing systematic CRISPR-Cas9 knockout screens in our cell lines. And so we are very fortunate to have a genome-wide CRISPR library that was developed by my colleague Kazuki Yusa at the Sanger Institute that targets 18,000 genes with 100,000 guides. And the way that we perform these screens is that we transduce the library into a pool of Cas9 expressing cells in such a way that we get about one guide per cell. Now, if that CRISPR guide targets a gene that's required for the fitness of a cell line, you get depletion of those cells from the pool over time. And you get depletion of the guide. And then we can use next generation sequencing and the sequence of the guide as like a molecular barcode to count the representation of a guide at the beginning and the end of the experiment. And things that are depleted at the end of the experiment are required for the fitness of cells. And so we can do this across many, many thousands of genes as depicted here, and we can do this across hundreds of different cancer cell lines. And so the data that I'm going to talk to you about today in more detail is the result of performing 941 CRISPR screens across 324 cancer cell lines. So each cell line was screened in triplicate. Now this covers a range of different cancer types as shown here, including things like colon, ovary, breast, and pancreas cancers. These tumor types were selected because they represent both some of the most common types of cancer, so there's a very large disease burden, but also cancer types where there's a real unmet clinical need, such as pancreas. Also important is obviously having the availability of a large number of cell lines for these screens. And so what we observed, I think, initially was very striking in that when we look across all these cell lines, we typically find about 14 to 1500 genes that are required for the fitness of a cell line. And in total, we actually found about 7,500 unique fitness genes required for the fitness of at least one cell line in the collection. And so this represents approximately 40% of all the genes that we tested have a fitness effect in at least one cancer cell line. And so we next examined how many cell lines were dependent upon each of those genes. So for example, some genes are actually required in only a single cell line, relatively unique events. Others are actually only required in a subset of cell lines, in what we call context-specific fitness genes. And we also have other fitness genes that are really required for virtually every single cell line that we've screened. And we call these common or core fitness genes. And these genes tend to be involved in very fundamental cellular processes, such as cell division. But what emerges is a picture of a high degree of diversity and heterogeneity in the set of cellular fitness genes across the panel of cell lines. And not only that, but given this very large number of fitness genes, it actually becomes quite difficult to determine which genes might actually represent good candidate drug targets for the future. So to nominate drug targets, we actually developed a target prioritization pipeline by integrating our experimentally derived fitness scores with multiple other data sources. And the goal here was really to be very systematic and unbiased to find the widest possible range of targets, and particularly new and innovative targets. And so the way we approached this is we identified genes that were required for the fitness of a subset of the cancer cell lines, these context-specific fitness genes. Core fitness genes were actually excluded from the target prioritization because we reasoned that they were more likely to be associated with non-selective toxicity in normal healthy cells. And what we did then is integrated these fitness scores for each gene with evidence, for example, that the gene is altered in patient tumors or aberrantly expressed. We also looked to see whether there was evidence at the pathway level that this gene was required for fitness. So were there other components in the same pathway that were also required for fitness in a particular cell line? And finally, we tried to identify genetic markers of differential fitness effects across our cell line collection. And we performed this type of analysis for each cancer type individually, as well as when combining all cancer types together. And finally, we also integrated this with tractability evidence to determine how druggable a target was for small molecule or antibody inhibition. And so what you ultimately get out of this is a ranked list of targets based on a priority score for each cancer type and pan-cancer, as well as their tractability for drug development. And altogether, we identified 628 candidate priority targets using this approach. This included, for example, well-established targets such as ERBB2, but many new potential candidate targets as well. The group 2 targets are particularly interesting because these are targets for which there's good evidence experimentally that they may be interesting, but in addition, there's evidence supporting their tractability for drug development. And there's also the group 3 targets, which again have good experimental evidence suggesting that they might be good targets, but at this point, generally have weak or no supporting evidence for tractability. And these actually tend to be enriched for things like transcription factors. Now, this is quite a busy slide, so we can actually look at this data on a per cancer basis. And this actual slide actually has an additional piece of information because it shows those targets for which we were able to identify a genomic marker of differential effect in the cancer cell lines. So this was a genomic event that could potentially predict a fitness effect if you knock out or inhibit the target. So in the setting of breast cancer, for example, we identified PI3 kinase as a very high priority target, as well as ERBB2 and ERBB3, other targets like CDK4 and estrogen receptor AKT, as well as PI3 kinase beta. And of course, this is very good evidence that this approach is working well because we're really capturing a lot of the well-established targets for breast cancer. But equally, we're finding many new things, and we're also finding things like some of the downstream transcription factors that we know are important for the proliferation of breast cancers. We can actually look at this data in a little bit more detail. And this just again shows the fitness effect in the breast cancer cell lines, but in defined molecular contexts. So for example, in cell lines that have an amplification of ERBB2, they have an increased dependence on ERBB2 as measured by this fitness effect. And that's shown here, where each circle is an individual cell line. Interestingly, we also see that again in ERBB2 amplified lines, these also are dependent on another EGFR family member, ERBB3. Unsurprisingly, we find that PI3 kinase mutant breast cancers have an enhanced dependency when you knock out PI3 kinase. But interestingly, we also see that when you knock out PTEN, which could also lead to the activation of the PI3 kinase pathway, we actually see a selective dependency on the PI3 kinase beta isoform, which has actually been previously shown. And we can find also new associations. So for example, here is a region of the genome that's recurrently amplified that contains these two known driver genes. And what we see is that cell lines that have this amplification are more dependent on KMT2D. Another interesting example is in the setting of colorectal cancer. So here, for example, we see targets such as BCL2L1 and MCL1 coming up. We also see things like KRAS and USP7. But very importantly, and just to note, we also identified Werner helicase as a very strong biomarker-linked target in this analysis. And I'll tell you more about that in a moment, but just to point out that we have this type of analysis for 13 other cancer types, where you can really look at this data in much more detail. So Werner helicase is a very strong selective dependency marker in cancer cells that are microsatellite unstable, or MSI. In particular, this is true for cells when you look across all cancer types together, as shown here in gray, but also specifically in the setting of ovarian and colorectal cancer. Now, Werner helicase is actually required for the maintenance of genome integrity, indicating that mismatch repair deficiency, which actually leads to this MSI phenotype, results in increased dependence on this Werner-mediated repair process. So I actually spoke earlier today in one of the other sessions about our work investigating Werner as a target in MSI cells. So I'm only going to very briefly summarize our findings again here. And briefly, what we see is that when you knock out Werner, this results in dramatic chromosomal instability and the accumulation of DNA damage selectively in MSI cells, as well as cell death. And as far as we can see, this is true for about 90% of MSI cancer cells. We've also used mouse xenograft studies to show that Werner knockout leads to suppression of tumor growth in vivo compared to control. So what we've done here is use a subcutaneous tumor xenograft model with an inducible Werner CRISPR in an MSI cell line. What we can see is when you have an established tumor, but then you actually ablate Werner through the use of through doxycycline-induced induction of CRISPR knockout, we can see that you get very profound tumor suppression compared to control cells. This also leads to loss of proliferation within the tumor mass and accumulation of apoptotic cells. And so these results and the identification of Werner as a potential target in MSI cancers has actually led to drug discovery efforts targeting Werner by a number of different pharmaceutical companies. And hopefully what I've been able to show you is using this type of approach with CRISPR screens allows us to identify many new candidate drug targets, including, I think importantly, a large number of new targets, as well as a wider range of candidate targets. So the data sets I've told you about are available through the Project SCORE website, which I mentioned earlier. And through the portal, you can access gene fitness scores across hundreds of different cell lines. And you can also mine for candidate oncology drug targets using the predefined criteria that we established in the study I've just shown you, but actually also using user-selected criteria. Now, since the publication of this study, we've really continued to expand and refined our analysis to identify new drug targets. And this work that I'm going to just very briefly tell you about is actually led by my colleague Francesco Riorio at the Sanger Institute and was actually done in collaboration with the Broad Institute. And so we've actually developed methods to integrate the independent CRISPR-Cas9 screening results that were actually generated at the Sanger and Broad Institutes. And we can bring these data sets together now so that we actually have coverage of over 16,000 gene knockouts across 786 different cancer cell lines. And so having this larger set increases our coverage across different cancer types, as well as the number of cell lines for each cancer type, giving us more statistical power to identify novel and cancer type and subtype specific targets, as well as new markers of gene dependency. And I've just shown you an example of that here. So what this shows is looking at dependency on MDM2 in lung cancer cell lines based on P53 mutational status. And if you just look in the Sanger data alone, you don't see any statistically significant difference in MDM4 dependency, rather, based on P53 status. But what you can see is when you actually combine all the data sets together, that cell lines that are P53 mutant have actually a decreased dependency on MDM4. A second example is looking in the setting of ERBB2 amplified breast cancers and whether they're dependent on the knockout of ERBB2. And here, when you only look in the Broad data, you don't see a significant increased dependency in the breast cancer lines that have an ERBB2 amplification. However, if you then combine the two data sets together, you can see that you see a very strong statistically significant effect. So we're very excited by these data sets. They're very new data sets, and we're beginning to mine them, as I mentioned, to identify additional drug targets. I'm also going to now tell you just very briefly about a separate study where we've actually integrated both the CRISPR and pharmacological data to interrogate cancer gene fusion function to identify new candidate targets. And I think this is just another interesting example of how you can use these functional genomic data sets. So fusions are used for diagnostic purposes, for prognostic purposes, and actually represent some of our best drug targets. For example, things like BCR-ABL inhibitors. But what's happened in the last few years is there's actually just been this huge explosion in the number of reported gene fusion transcripts. And this is really due to the advent of high throughput sequencing technologies. The challenge we face, though, is that for most, we don't really know if they have a role in cancer, and they could just be bystander or passenger events within a scrambled genome. And it's actually unclear which could be clinically informative. And so to try and address this last question, we've been using our CRISPR and pharmacological data to try and identify fusions that have a functional role in cancer cells. So we began this by performing RNA sequencing on our collection of 1,000 cell lines. And we used a metacaller involving 3 different algorithms to identify fusion transcripts, leading to the identification of just over 8,000 fusion transcripts across the cell lines. And what's shown in this depiction is each circle is a cell line and just shows you the number of fusions identified in a typical cell line, as well as some individual cell lines that have well-established or well-known fusion transcripts. For example, the EML4-ALK fusion that's typically found in lung adenocarcinomas. And of the 8,000 fusion transcripts we identified, about 14% of them contained a known cancer driver gene, and about 11% of them have been previously reported in patient tumors from an analysis of the TCGA data. So we next wanted to try and use our CRISPR data to try and determine which of these fusions might actually have a functional role. And to do this, we actually took advantage of the fact that most genes are actually targeted by multiple guides, okay? Now, in most studies, what you do is you actually take the average effect across all the individual guides for each gene, and that's what you report. But actually, what we did in this study is we determined whether each guide mapped to the portion of the gene within the fusion transcript, as shown in red, or outside of the fusion transcript. And we did this for both individual genes. And then what we can do is compare the relative fitness effect of what we call the mapping guides to the non-mapping guides by calculating a differential fusion essentiality score. And so what we can do is get this score for each fusion transcript within each cell line where we have these differentially mapping guides. And then we can use a statistical approach to calculate those that we think are statistically significant. An example is shown here in the AML cell line MOLM13, which has this very well-described KMT2A MLL-T3 fusion. And again, what you can see as depicted by these stars is we have both mapping on and non-mapping guides to the fusion. And this just actually shows the actual CRISPR data itself for each of the individual guides. This particular screen had 10 guides per gene. And what you can hopefully see is those mapping guides in gray have this negative fitness effect, whereas guides that are non-mapping did not have an effect. And so this type of example would lead us to the conclusion that this fusion is required for the fitness of this particular cell line. And we've done this now across all of our cell lines where we had and genes where we had a mapping and non-mapping guide. And overall, we could actually test about 2,800 individual fusions. And doing this, what we found is that only about 5% of all the fusions that we could test had a significant effect on cell fitness when we knocked them out. So we were able reassuringly to find many well-known fusions, such as the EWSR Fly1 fusion that's found in Ewing sarcoma. And for example, the EM4L fusion was also specifically required for the fitness of cancer cells. what I think is important here, though, is that for the vast majority of fusions, we didn't actually identify any positive evidence supporting a fitness effect. And in addition, many of the fusions which have been identified in patients and contain oncogenes were also not associated with a fitness effect. And this includes many fusions that were in frame and contain an oncogene. We did, however, identify rare, potentially actionable fusions in new histologies that we think are really interesting. So for example, we identified a BRD4 NUT-M1 fusion in small cell lung cancer cell line. Now this fusion is typically observed in a different cancer type called NUT midline carcinoma, where it confers sensitivity to a BET inhibitor. And indeed, in this small cell lung cancer cell line, we were able to confirm that indeed this cell line was exquisitely sensitive to a BET inhibitor. Similarly, we identified a breast cancer cell line that had a ROS1 fusion that's typically observed in lung cancers, and these cells were dependent or sensitive rather to a ROS inhibitor. And finally, we identified a RAF1 fusion in the setting of KRAS wild type pancreatic cancers that were exquisitely sensitive to a MEK inhibitor. Interestingly, though, we also found our spondin fusions, which are actually associated with activation of the Wnt signaling pathway in colorectal cancer. And these fusions confer sensitivity to porcupine inhibitors. But unlike colorectal cancer, these were not associated with increased sensitivity to porcupine inhibitors in esophageal carcinoma or cholangiocarcinoma. So collectively, we think that these are quite kind of interesting examples where you could potentially repurpose these drugs into new histologies. But they also point out that in some cases, there may be a contribution of tissue lineage, which also modifies that drug response. Like sometimes can actually mean that those cells are not dependent on a drug you may well expect them to be dependent on or sensitive to. Now, one of the more interesting fusions we identified was a recurrent YAP1 MALM2 fusion in 3 different cell lines from glioma, tongue, and ovarian cancer cell lines. Now, this had been previously reported in nasopharyngeal and skin cancers, but its function was not known. What we were able to show is that cell lines that had the fusion were very selectively dependent upon it for their cellular fitness, whereas other cell lines were not. Now, YAP1 is a transcriptional regulator of the Hippo pathway, and through binding to TAD1, activates Hippo pathway signaling. And MALM2 is a transcriptional coactivator involved in Notch signaling. What the fusion does is it brings together the transcriptional activation domain of MALM2 with the TAD1 binding domain of YAP1. And what we were able to also show is that the presence of this fusion protein is associated with really robust activation of the downstream signaling pathway. So this identified this fusion as a potential driver of the Hippo signaling pathway in these 3 different cancer cell lines. Now, we know that there's considerable interest from the pharmaceutical industry in developing Hippo pathway inhibitors. And our data actually suggests that these patients, although very rare, could be actually good candidates for these treatments. And so overall, using our CRISPR data, as well as mining for statistical associations of fusions with our drug response data that I wasn't able to show you today, we actually tested 3,354 fusions to see if there was evidence for them having a function in these different cancer types. What we found is that about 12% of them, we could find positive evidence. And so we think collectively this result really emphasizes the importance of ascribing function to fusions when interpreting them from genomic sequencing, because many actually don't have a functional consequence. We were also able to identify rare actionable fusions in new histologies, as well as identifying this YAP1-MALM2 fusion, supporting targeting of the Hippo signaling pathway in cells with this particular fusion. And so finally, in the last few minutes of my talk, I would just like to tell you about something that we call Project GROW. So we continue to use our cancer cell line collection extensively, and I think you've seen that through my presentation today. We actually also have a very active effort to generate a new generation of cancer models for functional studies. And this is because we recognize a number of limitations of the current set of cell line models. We know that they've adapted to 2D culture in very poorly understood ways. We often have little or no patient genetic data or clinical data to accompany them. And we know, and this is I think really critical, that we have poor representation of some tumor types and disease stages. And of course, if we really want to achieve precision medicine for the majority of patients, we need a set of models that reflects the diversity of the different patient populations so we can really study them. Now, historically, making new cell lines has been really difficult, often taking many months with very low success rates. However, there's been some very exciting advances in cell culture methods, and particularly the use of organoid cultures has dramatically improved the efficiency of deriving culture models. And so organoids are basically grown as 3D in vitro cell cultures using a basement membrane extract together with specially designed media that includes niche factors to support cell growth. And so it's now possible to actually derive tumor organoids representing different tissue types and molecular subtypes, as well as from different disease stages, with much greater efficiency than it was previously possible. So over 5 years ago, we actually reported a pilot study where we evaluated whether it would be possible to build a biobank of cancer organoids. We were able to establish a small bank of 22 organoids from colorectal cancer patients. And we were able to show that they really matched genetically the tumors from which they were derived and could be utilized for in vitro drug testing. And we've now performed similar studies in the setting of esophageal cancer, pancreatic cancer, and liver cancer. And based on these experiences, we've now set out to really build a much larger biobank of tumor organoid cultures, focusing on esophageal cancer, pancreas cancer, and colon cancer. I should add that these are all generated as part of an international collaborative effort called the Human Cancer Model Initiative, and all of the models are being made available as a community resource. So we've established a pipeline for organoid derivation at the Sanger Institute. We've created a network of 9 clinical sites to access tissue. These tissue typically comes from surgical resections. The samples are then couriered to the Sanger Institute, where we undertake derivation in a dedicated laboratory facility. And we then have very rigorous quality control, after which the organoids are banked and sent to the repository ATCC for distribution. They also then enter into our downstream analysis pipelines, including a genomic analysis, drug testing, and CRISPR screening. So to date, we've actually processed over 488 tissue samples from surgical specimens. And this has led to the derivation and banking of 136 cultures. Now, our success rate at 28% is slightly lower than what's reported in the literature, but we actually believe this is due to the fact that we take all samples received without any pre-screening, and that we actually have a very high bar in the sense that we have to generate a long-term culture that we can make 25 cryovials for a derivation to be deemed a success. Very briefly, just to say that we're collecting a lot of clinical data for each of the models So a total of 60 clinical features, including things like smoking status, disease stage, and prior lines of therapy. And of course, an important goal is to really have representation of the different molecular subtypes within this set of cancer models. And this just shows the breakdown of some major cancer driver genes in the first set of 32 colorectal cancer models. So for example, we have, if you just focus on PI3 kinase, we have cell lines that have PI3 kinase mutations, but coincident with KRAS P53 and APC mutations, whereas we have some others, that are only with P53 and APC. You have some that are just with APC, and of course you have models with just TP53. And so you want to have representation of all of these different cohorts. We also have representation of both microsatellite stable and microsatellite unstable tumors. Now, because organoids are grown as 3D cultures, this makes them a little bit more challenging to actually work with, particularly for some of the functional genomic screens that my lab routinely performs. And so we've been investing a lot of energy in developing new methods and reagents to improve our ability to work efficiently with these cultures. And so an example of this is that we've recently designed a smaller minimal genome-wide CRISPR library that uses only 2 guides per gene compared to 5 guides in our original library. And these guides were actually empirically selected by mining the data from the Project Score screens I presented to you earlier. And so this library is actually 60% smaller than the original library and 42% smaller than any other genome-wide CRISPR-Cas9 library. Now, importantly, we've been able to show that this new library retains specificity and sensitivity to identify gene dependencies compared to the original library. Both here in cell lines, you can see there's good correlation in the gene dependency effects, as well as when you look within an organoid culture. And so we believe that this smaller library is particularly useful for when working with things like organoids, where it's more difficult to grow cells, or actually when measuring really complex phenotypes, for example, using these for a flow cytometry-based assay. And so kind of bringing all this together, our long-term vision is to build a passport for each of these organoid models. And this will include things like patient and clinical data, for example, previous lines of treatment, genomic data sets, for example, comparing the organoid back to the original tumor, as well as a description of driver alterations that are present. And then, of course, functional data as well as part of our cancer dependency map. I should also add that the organoids are actually, as I mentioned previously, being made available through the public repository ATCC. And in fact, there are actually 100 organoids that are available to purchase from ATCC currently available. And we've also then created the Cell Model Passports database, which is a hub for information on preclinical cancer models. And this actually includes genomic and functional data sets for over 1,900 cancer models, including both cell lines and organoids. And using the Passport website, it's very easy to search or filter for models of interest. So in this particular example, I have listed all of the organoid cultures which are currently available in the Passport database. You can then click through on a specific model where you can access all the patient clinical data, for example, for that model. You're also able to access the genetic data that we currently have available. And there are also links to ATCC where the organoid can be purchased. Now, the available data sets for organoids are currently a little bit sparse, but obviously will be expanded over the next few years. So finally, I'd just like to close by thanking all the many people at the Sanger Institute involved in this work. It's very much a team effort, as well as a fantastic group of collaborating laboratories that have been involved in this work, as well as my funders. And finally, I would very much like to thank you for your attention. Thank you.